My name's David Aanensen. I'm a Professor of Genomic Epidemiology at the Pandemic Sciences Institute at the University of Oxford. I'm Director of the Centre for Genomic Pathogen Surveillance, and what we focus on is a combination of genomic epidemiology, understanding pathogen dynamics from whole genome sequencing, engineering and building tools that help others to understand the information that comes from whole genome sequencing, and a real focus on capacity building for genomics in largely low and middle income settings. How does this technology and information get owned by locations that stand to benefit the most from? It's a huge honour to be awarded the Translational Microbiology Prize. It's a real testament to the team that we've been working with for many, many years, and our collaborators, both past and present. It really sort of justifies the work that we do, and it's a real honour. So with respect to infectious diseases, we really need new tools in our armoury on how to spot and stop and prevent infection spreading. We have a very strong interest in antimicrobial resistance. It's a global problem. And genomics allows us to look at the population of the bugs with precision to try and think about how we can actually intervene, to either develop better treatments or new treatments or keep treatments relevant, but also things like developing vaccines and developing targeted interventions that we can deploy in our fight against infection globally. We saw this very strongly in the pandemic. Genomics, really for the first time, was used as a frontline public health tool. 30 million genomes were sequenced globally. That's quite astounding. And it meant that we could look at the population of the pathogen and break it up into components like alpha, beta, Omicron, and understand that Omicron was worse than alpha. So can we use precision diagnostics, precision treatments to go after subsets of the population? Now, we feel very strongly that genomics allows us to do that for antimicrobial resistance, but one of the key things we need to be doing is surveillance everywhere, in as broad a spectrum of areas as possible, because these bugs are different in different parts of the world at different time points, and understanding that, genomics allows us to read an evolutionary snapshot of that bug in that point in time, and that's a transformation and very exciting tool that needs to be democratized. So I worked in the web industry for a couple of years after my master's. I did a master's in neuroscience, so it was molecular biology. Worked in the web when the web was really exploding. I landed back in science for my PhD in a lab that was focused on molecular epidemiology. I was lucky enough to work in the lab of Brian Spratt, combining genome sequencing, and this was Sanger sequencing of 7 genes to be able to identify strains of bugs. And I was very interested in how you show stuff off via the web. So I got to combine my two loves, right? Technology and then also genome sequencing. And genome sequencing expanded into whole genome sequencing, and the web started to evolve, things like Google Maps, Android phones. So I've been able to kind of sit in the middle and kind of drive this delivery of both digital information and then the web on the other side to combine those two bits together. I was lucky enough to be exposed to the European antimicrobial resistance surveillance system. And that many countries across Europe every year collect the amount of proportion of resistance to certain antibiotics, to certain drugs. And this information was used to produce very nice maps, heat maps, that show increasing resistance over time. But of course, if you're increasing resistance to one drug, to one bug, genomics gives us the precision to say, is that one clone, or is it multiple clones, or is it a variant that's coming from elsewhere? So it's really quite obvious that the application of the kind of precision you get from genomics in the area of public health was going to explode. And we're seeing that now. So I got very, very passionate and interested in how can I continue to show off via the web, but also bring this new technology that was incredibly exciting, being able to understand the entire genomic complement of an isolate together. And that's really been the drive to how do we actually scale this up and bring it elsewhere. Knowledge sharing is important in science, it's important in all areas of research, and that goes, it's a broad spectrum. That term encompasses a vast amount. So, if I think about data sharing within genomics, I mean, it's very important to have as much sharing of genomic data that's been sequenced globally, right? Because that gives us a much better ability to get on top of what's happening. But there's all sorts of political issues around sharing. There's been some quite detrimental examples of where data have been shared from countries and then used to develop products to sell back to the country. And that we have to work out best ways to democratize the value, but also have the value come back to locations that generate the data. And increasingly, that's going to have a profound impact on our capacity to do national and international public health using this kind of data. but it's incredibly important. So I think models have to be built that enable local ownership of both the interpretation of the information, but also the value that comes from that sequence information. A lot of good work being done across the world to try and work out how that happens. From a technical point of view, I think sharing information is all possible. We have a globally connected platform that allows us to deliver data upwards and downwards in information chain for decision makers at all sorts of levels. So I don't think much of the challenges at the moment are technical. There's a lot of challenges around sharing information about the infrastructural setup, so actually standardized protocols for doing so. But I think it's a practical challenge now. Much of the engineering is in place, both from the data sharing perspective and also the technology perspective. It's actually implementation. How do we ensure that the benefit of these technologies get to the locations that can own them the best? The moments I'm most proud of are being able to click with members of the team and really see new ideas and new things come to life. I'm very lucky to work around a bunch of really talented individuals, scientists and engineers. And I get some immense satisfaction from both. And actually both come together in the middle. So our engineers know a vast amount about microbiology, infectious disease epidemiology and pathogen surveillance. And our scientists know an awful lot about JavaScript and how to deliver information and visualization. Because actually it's a very visual picture, and what we need to do with this side is bring it to life in a way that's understandable by somebody who needs to make a decision. So bringing those two together and sitting in the middle, I'm immensely proud of the fact that we're doing that, and we're bringing folks on both sides into this middle stretch, which is where translation comes into action, right? Where you feed from both sides. You have scientific discovery that goes into your engineering translation, and the translation of the engineering and understanding that side feeds into questions that we have in the scientific bit. So I'm terrifically proud of that. I mean, I'm always, getting a grant, those feelings are immense, but actually working with partners in countries that we have and actually just seeing that actually some of the stuff that we're doing is making some kind of benefit, that's, how can you not be terrifically proud of that? And that's as a consequence of the team coming together and really sharing a passion for delivering. I firmly believe that we want to see the benefits of science in society. And also it's incredibly satisfying to see something that's come from a group that you're involved in that then gets picked up and used by multiple people. And this is where, again, very personally, the web is a very good platform for sharing. And actually some of the data tools that the team have produced over the years are used globally, in public health labs, but also for all sorts of endeavours. That's tremendously satisfying, but also you can see the impact it makes. So I think actually the purpose of certainly my focus on research is that there should always be a translational aspect, that we should do our science thinking about how we can benefit the human population and animal population, but planetary health, One Health, whichever kind of phrase is necessary. Because that's the translational purpose of what we do, and that's the funders appreciate that, and it's important. Brilliant, fantastic. Thanks, Duke, for the introduction. Yeah, it's been an interesting set of years, but it was a pleasure working up at the Sanger for a period, and now at the Pandemic Sciences Institute, actually. The Big Data Institute at Oxford has now transformed into the Pandemic Sciences Institute post-pandemic, which is a real pleasure because we have a group of people who really drove and developed the COVID vaccine, analytics, modelling, etc. So it's a really, really, really great pleasure to firstly accept this prize. I've never won anything before. I won a school raffle when I was about 9, and the prize was a box of soap. I mean, what use is that? Anyway, so I'm going to talk about sort of two things that I was lucky to be around the evolution of. One is the next generation sequencing, so the transition from Sanger sequencing all the way up to now being able to sequence the whole genome. And then also the evolution of the internet. So I did a master's in neuroscience after a degree in biochemistry. Then I worked in the web industry in around 2001, just before everything crashed. And if anyone's seen Nathan Barley, it was like working there. I started on day one, had never built a website before. They said, here's a team of 2 people and here's Alfa Romeo. Go and build the Alfa Romeo website.
Oh, and by the way, there's a sports car around the corner so that you can get the feel of what Alfa Romeo's like. So we were in the middle of Soho in this very exciting period, and the web really sort of uniquely allows you to show off in a way that many other technologies don't. So over the years, as the web progressed, and as whole genome sequencing progressed, I started my PhD with Brian Spratt at Imperial around MLST, so I was building data systems for multi-locker sequence typing. Those two parallel things, we've kind of sat ourselves in the middle, really, and it's been a real pleasure. So anyway, my grandma sucks eggs slide, this is for everybody knows all of this anyway, but one of the things that we try and do in infectious disease epidemiology is really understand for an infection, are there related cases, and can we use that information to do something about the related cases? Where are cases of a disease? What are they infecting, when, and where? That could be within a hospital, could be on a farm, the environment, water, plants, food production plants, and we want to try and identify whether the things are related and whether we can stop that. And that scales up to between organizations and institutes nationally, internationally, and ultimately globally. And you know, as we're all well aware, we're a very, very connected world, so this is very important to try and understand this information. And one of the key tools that have been developed is using genomics. So you take a bunch of samples, you run them through your favorite genome sequencer, and then you can relate the similarity of those samples to try and understand whether things are related or not. And because we have the whole genome of these samples, we can look for markers in those genomes that may indicate a heightened risk. Antimicrobial resistance, for example, the presence or absence of mutations or genes. But there's a whole bunch of information there, right? So you can imagine collecting loads of information about this, lots of complex information here. How do we distill all of the information that we could get from the epi, the real world, into something that combines the power of genomics to deliver to this individual here to try and interpret and actually make some decision, do something about it? And that person could be sat at any one of these levels, right? It could be somebody in infection control, could be somebody at the UK HSA, and every country has a national entity such as this, so this is scalable anywhere. In continental bodies like ECDC, and ultimately upwards towards the WHO. So we've spent a lot of time in this space really thinking about how do we research and understand the evolution of the genomics, but also then combine that into these kind of deliverable tools. So genomic epidemiology. So you take your samples, you run them through your not to scale, and also other genome sequences are available. I'm a big fan of PacBio, but short read sequencing over the years, nanopore, portable. You run your samples through and you get a whole bunch of genomes out. Then the challenge is, what do you do with that information? So let's say we've sampled and sequenced three things. We can compare the genome sequences to indicate that in this case, 2 and 3 are more similar to each other than they are 1. So this kind of tree-based representation is what we use to indicate that 2 and 3 are something more interesting than 2 between 1, etc. And then let's say we start mapping some kind of parameters. It could be epidemiological, it could be presence of genes, it could be resistance to a drug against this kind of structured relationship. So in this case, 2 and 3 are similar genetically, but also they have these 4 things in common as opposed to 1. And then let's say we've got geographic information. So in this case, 2 and 3 were sampled very close to each other, 1 was further away, etc. You can see we're building our hypothesis around 2 and 3 being important, the relationship. And if we've got temporal information, they've both been sampled in the same point in time. So these kind of methods of bringing the information together and building our hypothesis are what we do all the time in genomic epidemiology. So this is a real-world example. This is epidemic MRSA 15, sequence type 22, which caused most of the MRSA in the UK. This is the tree. The red bits represent that these are methicillin-resistant MRSA. The greens are MSSA. And you can see geographic clustering here. So there's a cluster in the UK, one in Germany, and one in Portugal. And we know through research that MRSA did emerge in the UK, and it was as a consequence of the introduction of fluoroquinolones in the mid-1990s in and around Nottingham. And so this represents independent introductions outside of the UK into Germany and Portugal. So these are the kind of ways that we look at this information on a routine basis. And these trees are a very good visual way of representing the relationships between samples. And the trees can be viewed in different ways. So this is the same tree, just viewed in different. So the relationships haven't changed, but it allows us to bring our visual acuity on top of seeing and pulling out the relationships. So this is a classic, what we call a comet-like shape. This is kind of an outbreak, very rapid dispersal. These are the background information, and actually this is incredibly important. Having good context, good standard information that you can compare against to be able to see whether something's emerging. So the other thing is these bloody things evolve. So we've got to keep doing this continuously. And can we use temporal information over time to try and understand how a population of the bugs are changing? And whether, for example, you can see here, this is a very terrible cartoon, but the green samples are increasing in frequency in the population. And you know, if you think of SARS-CoV-2, we all now understand very well that a variant emerged and took over the population. Bacteria and other things are a bit more complicated than this, but you can use this information to, for example, develop diagnostics against green, and could we develop interventions against green, etc., and monitor those interventions over time. So back to some of the stuff that we've done. So we've spent a lot of time developing data applications that are really focused on what I've just described. How do we best collect, represent, visualize, process, understand the information by doing research and bringing that back into the engineering for these applications, focused on supporting very local information and then thinking about how we can place that into larger context. So I'm going to indulge myself a little bit and show you a couple of these tools. The first one is about collecting good quality metadata. So I was in the lab of Brian Sprout when Google announced Android. This was back in 2008. And it was very clear that we could use this kind of mobile tool that had access to location, photographs, etc., to do something that would allow us to collect information. I was actually working on with a colleague who was focused on frog fungi, Batrachochytrium dendrobatidus, if anybody knows this. This was a decimating amphibian species across the globe. And I got quite excited because you can put a latitude and longitude on a frog in a way that you can't a human. So and there was a real need in the herpetological community to be able to collect spatial information about the amphibians. So we mocked up an application. We submitted it as one of the very first Android apps, actually, and then have over the years progressed this. So I'm going to show you what it does. So the idea is that you can build a data collection form, load it onto loads of phones, collect the information, centralize the data. So let's build a project. So I'm going to call it MicroSoc, all lowercase, and a small description. Are you enjoying the conference? And I'll give it a form, which is form 1. Okay, and we're going to make this public because we want anybody in the world to be able to submit information and collect it. So I'll create that now. So the first thing we have to do is build a form. Now, this left-hand side contains all the kind of text-based things that you can imagine putting into a form. And then there's a bunch of media questions at the bottom, like a GPS. So anything you could get access to on a mobile tool. So let me drag a text question. What is your name? And we will not require that. But let's go. Let's do a drop-down. So this allows you to define the choices, so you can control the type of data. Are you enjoying the conference? And then you define your answers. Yes. Add an answer. Absolutely yes. Brilliant. Okay. Then we'll add a. So you know you can think of any kind of question that you want. So then we'll add a location. So this is take your position, and then we'll add a photograph because everyone loves a photo. Take a photo. Right, now, so if I save that project, that's kind of essentially ready to go. But you can see the kind of text questions. You can build in lots of logic. You can have splitting forms. You can have multiple forms per question. But if we now go back to the main page, so now I have my own home page, and I can maybe upload a logo. Let's imagine that I very carefully selected the Microbiology Society logo last night and saved it. And then we've got a home page now, right? So this home page is where all the data is going to come in. So if I then switch to my phone, which I have here. Okay, so the app is available on Android and iPhone. So I tap EpiCollect, And what I need to do is now add a project. So I can just type in microsoc, and this will pull up the Microsoft project that we've just created. There's a little logo, so I load it onto my phone, and then I go into it, and I can add an entry now. So these are the questions I just defined. So my name is David, let's do that. Here's the, are you enjoying the conference? Absolutely. Next one, take a GPS, so it takes a GPS now. We're inside, so the accuracy is pretty terrible. You can update the GPS, but you see the 85 means that there's an 85 meter kind of radius of uncertainty around, but you can change that. And then take a picture. So I don't know whether everybody can wave. 1, 2, 3, wonderful. There we go. Let's use that photo. So then I'm done. So I can then go on and move and get my next answer, and update that information. So what we then need to do is upload the information. So this will upload the text first, because you don't need much internet to upload the text, and then you can upload the photos separately. I should say that you don't need any internet to be able to collect the information. You only need it to load the project initially and then submit the data. So that's all done. So now we can go back to our home page and refresh, and there's one entry, and I can view that information. There's the data, there's your real-time kind of close to useful picture, some nice smiles. There's my daughter and other half and my mum and dad on the front row. That's always good. And then you have a map. So there we are. There's your information. So that's the basic premise. And if you go to the website, there are hundreds of thousands of projects that people have picked up with all sorts of activities. So we built this for epidemiology, but it's been used for vast amounts of things over the years. Citizen science, environmental science, schools, field courses use it. I went with the University of Bath for annual field courses, and instead of notebooks, they now have longitudinal samples. It's really, really taken off. So what I've done is I've downloaded about a year's worth of data. So there's about 150 million data points over the last 10 years. This is a snapshot of about a year, and this shows about 1.6 million data entry points, and you can go and zoom in anywhere in the world. This is Mexico City. So every dot here represents somebody taking some kind of information for a particular kind of project, and you can go and view things like this is in Colombia. Each one of these dots, these are vaccination campaigns. They have rollouts for Aedes mosquito. So there's a lot of public health projects, but there's also lots of other non-public health projects. I mean, I can spend way too much time looking into this. So this is interesting. This is Zambia and Zimbabwe. So all of those dots represent projects that people have collected with EpiCollect. And you can go all the way down to individual information here. But we were like, what are these lines? So these lines actually represent the road systems. So the Zimbabwe National Electricity Company uses EpiCollect to monitor the health of electricity poles. So they'll send people out with a simple questionnaire, voltage, etc., etc. So you can see the sort of vertical applications of this system. So that's EpiCollect. That one worked, Duke. We're halfway there. So I described this as being a general paradigm in genomic epidemiology. So I was doing much more of this kind of study in here. So how do we kind of provide a really simple way to get this sort of information from everybody who's using this kind of collecting geographic information, etc. So a second tool that we developed was called MicroReact, and this was about interactivity with that sort of information. Once you generate your genomes and your tree, and you've got all your other epi information, you really want to delve down into it. So MicroReact allows us to do that. So we have, again, a website, and we develop these tools by thinking about things that are appropriate horizontally that can be applied vertically. So there's a bunch of example data sets, for example, the West African Ebola epidemic, nice, which essentially it just places stuff on a map. You've got your tree, you've got your temporal information, and you can interact with this. You can, roll along and see. You can select information on the map and see where it is on the tree and temporally, and that's kind of really the very simple basic premise. But we want to make this easy for people to be able to build their own. So let's say that I have in my lab. I've taken some samples and I've genome sequenced them, and I've got a file which contains some information about those samples. So here's a metadata file that I've got a latitude and longitude. I've collected that with either GPS or you could use EpiCollect, right? And I've got a year, month, and day of sampling. I've got where that's been sampled. And in this case, I've tested some antibiotics. We've got resistant or sensitive. So that's a set of information about the samples that I've sequenced. And then my bioinformatician has nicely produced a tree. Now, this is, nobody ought to be viewing this kind of information, but this is the industry standard way of describing a tree. It's a Newick format, and it's essentially a bunch of concatenated brackets that describe the branching point, and it gives us an ability to visualize that information. So I've got my two files. I'll upload those here, and this is me investigating an outbreak. So what this does is the system interprets those two files, plots all the geographic points. So oh, look at this, where are we? Oh, this looks like a terrible outbreak. What's going on? So there's a cluster of points around this particular location coinciding with this time point, which I think was Richard Lenski's opening lecture. Maybe it was everybody in the same place, but you've got your tree. And again, everything's kind of linked all the way through. But we can see quite clearly here that there's a branch that's very similar. So let's add some of that metadata that I described. So let's put the resistance information. So then you can see here that this branch, everything's resistant, and the others have mixed sensitivity. So if I was investigating this, I'd be most interested in this particular cluster, right? And I'd want somebody, let's say I wanted to hire this up the chain, I'd want somebody to be able to see it. So I'm going to add a little note to this system saying, have a look at this, potential outbreak, etc. And what I'm going to do is I'm going to save that as a new project. And then what I'm going to do is I'm going to email that to my colleague who's higher up in the infectious disease public health chain, who'll be able to jump straight back into that particular location and see that particular branch on the tree. So we wanted to not only have this as a way of displaying and interacting with the data, but to think about the public health utility of using this kind of system. So this is an example of it being used very early on. I was part of the COG UK initiative, and Dugan, there was a meeting, a very infamous meeting at Wellcome, where we all got together to try and work out how we would do it. And this was the very first report that we sent to the government. So this was the very early on in the pandemic, was the information about the number of genomes that had been sequenced at that point. So here we've got just over 1,000 genomes, and if you think there's like 30 million globally now. So this was the very initial report that described the fact that there were different variants in the UK. These proportions were showing the different variants. So this went upwards to SAGE, and then that information was used to try and think about intervention. And then we saw this kind of really explode. So these are national level instances of MicroReact, where countries took this on to be able to display the different variants that were emerging in the pandemic. Here's the USA, Argentina, Spain embedded it within their national reporting. This is a proud moment. That's Jacinda Ardern in New Zealand. New Zealand did a very good job. I'm very happy that she got to use MicroReact because most of my family live in New Zealand, so it's a real pleasure. This is, we have now worked with public health agencies to have this installable, so this is used on a routine day-to-day basis to interpret public health activities in many, many US states. There's an instance in the ECDC, et cetera, et cetera. So it's being used for kind of frontline public health support and decision-making. And we had quite early on a partnership with MGen, microbial genomics. So if you submit a manuscript, you can upload your MicroReact, so it gives you access to the raw data. So these are the kind of ways that we've moved this forward. Okay, so rolling back a little bit to genomic surveillance. So this kind of longitudinal ability to understand the variation in the evolution of bugs. Why do we do this? Well, we can map the pathogen landscape. We can use this information to identify trends and changes, to monitor emerging variants and clones. And this is quite clear that during the pandemic, this was transformational. And this can help inform knowledge gaps, application of infection control, treatment, and ideally vaccine design and rollout and monitoring. And this is appropriate for any pathogen, right? It's not just SARS-CoV-2. We have a particularly strong interest in AMR because we're all going to be affected much more over the course of the next number of years. And the WHO has published a very nice set of priority bug-drug combinations that are really our highest priority to really understand what's happening with the emergence of antimicrobial resistance and how do we actually work towards interventions in affecting this. The WHO has also set up for global surveillance quite a nice simple framework where the idea is that every country has a national coordinating and reference lab that's hooked into a set of sentinel sites, hospitals, that are collecting phenotypic information. So this is the link to the patient. Information goes upwards here, and then ultimately into the global antimicrobial resistance surveillance system. So this is GLASS. It's a really simple framework, right, for describing a country. And this is kind of the current picture. Now, countries, member states are in very different levels of maturity within here, depending on where you are, UK versus Nigeria, for example. But the current picture is really focused on AST, antimicrobial resistance, susceptibility. So back 15 or so years ago, I was lucky enough to start interacting with a chap called Hajo Grunman, who established our European antimicrobial resistance surveillance system. And this was based on countries submitting on an annual basis proportions of resistance to particular bug drugs. So, and then over time, you could do these fairly simple but quite informative maps that indicated increase in resistance. So this is proportion of invasive E. coli isolates resistant or intermediately resistant to third-gen cephalosporin. So this, this model of national and sentinel, that's exactly how this information was captured. So I got very enthused by Hajo's enthusiasm for delivering on public health, and he had a particularly strong interest in the emerging field of genomics, and it was kind of one of these opportunistic meetings that led us to kind of come up with a project for leveraging these national reference labs and sentinels to do some genomics. So we suggested that a project involving 430 hospitals from across the region would focus on bloodstreams infections and collect the first 5 methicillin-resistant Staph aureus and then a matched methicillin-sensitive Staph aureus. So we'd be able to use genomics to map out the population structure. And then can we think about ways to deliver that information back to all of the member states? So that kind of led to some extent to MicroReact, so it was one of these democratization of the information. And then we published the first kind of European snapshot of what Staph aureus looks like. So this is the tree showing all the major and minor clones. This is that MRSA 15 snapshot that I showed you earlier, and it really serves as a baseline for defining context going forward and allowed us to describe with precision what the population looks like. We could also do, and I hate when tables are put in presentations, so please don't read. But one of the big promises of genomics is being able to predict phenotype from genotype. Now this table, essentially we had national reference laboratories who phenotyped all the samples. They were all sent to UCAST, who sets the breakpoints, so they phenotype, that's the gold standard phenotype, and then we had the genomics. So we could do this 3-way concordance to see how concordant the relationships were. And essentially across most of the frontline antibiotics for staph, the genotypic predictions were as good, if not better, than a heterogeneous network of labs doing different phenotypic tests when compared to that gold standard. Now, staph's pretty well behaved. We sort of know that the concordance is pretty good genomically, but when you go to other bugs, like this beast here, Klebsiella pneumoniae, it's very different. The Klebsiella evolves very differently. You have plasmids, you have very complicated mechanisms, multiple mechanisms for the same resistance. So our next step after staph was to think about Klebsiella. So with Hajo, he instigated the UScape project, and we again did all the sequencing. And Sophia, who's in the audience, is a very talented scientist, and we're lucky to have in the team, describes the population structure for Klebsiella across Europe, all the major and minor clones, where the mechanisms were. And I'm going to encourage you to read the manuscripts rather than me going into the details. Sophia can do this much more justice. But we also then sequenced using long reads plasmids from about 100 of these samples using PacBio. So plasmid epidemiology, which is going to become important. But again, back to that kind of evolutionary temporal change, a second survey was undertaken in 2019. And this then allows us to look at the bits in the middle. What changed? And in Klebsiella across Europe, there's been an increased acquisition of carbapenemase genes. We would expect that within high-risk clones, expansions of multi-drug-resistant clones and isolates with increased virulence. And this information can then be used to trigger action. So the ECDC, seeing that there was an increase in this particular sequence type, then launched a further investigation in Greece to try and understand what the epidemiology was of ST39. This is not directly out of the surveys, but the kind of alerting that ECDC does on these kind of molecular level clonal based, so ST23, a really nasty hypervirulent Klebsiella, alerting that led to a diagnostic for other countries outside of Europe to be able to identify ST23. And then there are a number of companies trying to develop vaccines against clones, so mRNA type vaccines, which is where it moves across. So, okay. All very well and nice. I'm going to step now back a little bit to address some of the challenges of processing that data. So these trees are wonderful, but it takes a long time to produce through bioinformatics pipelines. This is a massive challenge, bioinformatics, right? If genomics is in lots of labs everywhere, you can't require a bioinformatician in every lab to be able to do this kind of public health interpretation. Anybody who has sort of grown up with bioinformatics has written a pipeline themselves. If you look in every public health lab in the US, there's different, but it's a mess, right? And we need standardization. And we need it to be made easy for anybody to have a genome sequence to go all the way through to that kind of interpretation as rapidly as possible using gold standard research methods. And we've got to think about this as being embedded in empowering local labs to be able to maximize the value of the global data to deliver locally. I'm just going to point out these. My daughter's in the audience, so she's counting the number of unicorns that were in the presentation. I borrowed this from Nick Lehman. How many so far? 2. 3. Spot on. Okay, so the last one I'm going to jump out to is Pathogen Watch. So what we wanted to do is to think about how can we provide easier access to bioinformatics so that if you are in a lab, you don't necessarily require a bioinformatician to be able to do all the data processing. So, in this case, in my lab, I've sequenced a whole bunch of genomes. So this folder now contains a set of genome data. So each of these files contains an assembled genome. So it has gone through the assembly process. And then for each of these genomes, I now have a metadata file that, again, contains a bit of information. It's got a lat long, a day, month, and year, and essentially anything else we want to help understand the information. So Pathogen Watch, we have all of the genomic data that's out there, and I'll touch upon that in a minute. And what I'm going to do is upload all my single genomes. So instead of getting my bioinformatician to do this, I drag the information into the browser. And what this is doing is it's uploading the assemblies to a server, and the first thing that's happening is that server is predicting what the species is, because agnostically those ACTGs, so there's a big reference set of genomes, and then based on identifying what the genome is, it's running a series of set analytics that are based on community standards for what the right analysis methods are. So that's finished. So it's identified that in those 16 files I just uploaded, there's one Klebsiella pneumoniae, and then these are the analytics that are undertaken. Anybody who does Klebsiella genomics knows the tool Kleborate from Kat Holt's team. So essentially this runs Kleborate in the background. Kleborate's a command line tool, but we want to make this really super simple to plug in the right analytics. 12 Staph aureus genomes, and for Staph it's predicted the AMR, MLST, etc. And then there's 3 SARS-CoV-2 viral genomes that I slipped in to keep the viral folks happy. But it's agnostic, right? The whole process is, if you've got an analytic, you can embed the analytic in whichever species. So here's the table of information. So each genome gets a report. So let's open up the Klebsiella. So Klebsiella MLST, that's a typing methodology. CG-MLST, it's predicted the surface polysaccharide, which is important for understanding potential utility of vaccines. So being able to predict whether there's a K or an O antigen and what type it is very important. The antimicrobial resistance, so this is presence or absence of mechanisms, et cetera, and plasmid typing. And then, so we have a whole bunch of Staph here. So what I'm going to do is take this a little step forward. So each genome does get its own individual report, which contains everything, but I want to get to the point where I can try and understand the similarity between these genomes. So I'll call it microsoc again. And now what this does is it's going to build a tree. So it's going to look at all the shared sequence information across all of those Staph aureus genomes, count the number of differences, and relate that as a tree. So that's, again, finished. And because we've got our lat longs, it's placed it on a map. We've got the temporal information down here. And because we've run all the analyses, we can then overlay some of those analyses that have already been run. So we ran an analysis to predict the resistance to particular antibiotics in this case. So this is the presence or absence. So then we can choose one and map it onto the tree. So here we've gone from having a set of raw genomes, thrown everything in, to coming up with a hypothesis around a similarly geographically located set of samples that are genomically similar, that have a particular resistance determinant found in the genome. Okay. So, just then touching on, so we spent a lot of time building these kind of horizontal applications that are applied vertically. We spent a lot of time doing sequencing and trying to interpret that information, and the team of scientists, we look into this information how to build in. But actually, how do we take that out into non-sending samples to the UK? So I was at the Sanger for a period of time, and those European surveys, samples were sent into the UK, we sequenced them, and then the information was sent out. But actually, this technology is cheap enough to be able to implement in any location in the world. So we applied for an NIHR project, got a big 4-year grant to essentially work with partners in some of the nicest places in the world to travel. It's a real struggle sometimes to visit the Philippines and Colombia, but we work with partners here to try and look at, can we develop horizontal technical support packages that can be applied in different settings to embed genomics in national AMR control programs in this kind of framework. So we work with, lucky enough to work with a very inspiring team in very different institutional circumstances. Ravi works for a teaching hospital that's embedded into lots of hospitals across India. Sonia works in the Department of Health, the RITM in the Philippines. Iruka works in the University of Ibadan, but wrote the National Action Plan for Nigeria and works very closely with the Nigerian CDC. And Pilar in Colombia works for an Agrosavia, a company called Agrosavia, who are a public-private partnership that focus on One Health. So with this partnership, we developed a set of technical support packages, standardized process for doing the lab, DNA extraction, phenotypic identification, VITEC for AST testing, standardized bioinformatics to run the analysis, standardized data flow for linking information to that kind of epidemiological data, and then support for financial processes within each of those organizations, training and into IQA. And there's a special edition of CID that you can read that shows all of these kind of support pieces. But essentially, each of those sites now has a functioning autonomous genome sequencing facility. So how's it being used? Here's an example from the Philippines. So Celia Carlos, who was the director, really bought into the power of genomics. And actually, we were presenting for a couple of years, and it was only after 2 or 3 years Celia said, I think I kind of can see it now. But had a real foresight to understand that this was going to be transformative. So in the Philippines, they have a 30-year phenotypic surveillance system, which includes sentinel hospitals across the region that submit that phenotypic information upwards on an annual basis that goes into a report. So it fills within this structure here. So those phenotypic profiles for a series of antimicrobials are used to collate and inform local hospital information upwards to GLASS and at the Department of Health for treatment strategies. And this is what the report looks like. So every year, this is proportion, resistance, between years going up or down. So this is Neisseria gonorrhea for 3 antibiotics. But actually, what on earth does this line mean? Genomics will give us the power for precision to indicate, is that one clone? Is it one variant that's come in? Has it come from another country? Is it homegrown? Does it have multiple resistance determinants? So this is one of the things that we wanted to do when we embedded information and genome sequencing. So the Philippines at the RITM now have both Nanopore and Illumina, and when they sample and sequence, they can use this information to describe the output with respect to what's the strain, what's the sequence type, what are the resistance determinants, and whether those resistance determinants are delivered on a vehicle. Is it the plasmid or is it the chromosome? So that information gives them much more precision for public health implications. And this is a manuscript where they described using and leveraging the phenotypic information. So as those phenotypic profiles come in, they're scanned for clusters, and then genomics is used to define whether that cluster represents an outbreak or whether it's a different set of samples, much back to those first slides. So this is a manuscript where they described identifying a cluster that identified a long-term intensive care outbreak of Klebsiella pneumoniae, and then infection control measures were put in place. So it kind of leverages information, maximizes the use of small amounts of funding to do genomics, and this has now been taken up and funded by the government. So this is a routine thing that happens on a regular basis. So it really does leverage genomics on top of standard phenotypic surveillance. And then our other partners are using that kind of process to look towards building within. So Iruka, again, a very inspiring individual, has really built the national capacity in Nigeria, along with the Nigerian CDC, who looks to the Philippines for using these standard kind of processes to apply vertically. Okay. So as a consequence of that project, we were very lucky enough to be designated as a WHO collaborating centre, and this really enables us to take the kind of understanding of the implementation of genomics, the standardised process and protocols, and really use that to try and encourage member states or deliver information for standards across the whole board. One of the things that we do within the team is think about this approach across the board. So can you develop something horizontally that can be applied vertically in lots of situations? And that applies to the engineering. If you bring a function into a tool, could you imagine it being used for the Zimbabwe Electricity Company, for example? If you have a protocol or genomics, can you envision it being used for pathogen A, B, C, D, or E? Okay, so there is a bit of an elephant, which is the big trees. These trees get bigger and bigger and bigger and bigger and bigger. It's one elephant, darling, not a yeah. So here's a big tree. So this is in COVID. So again, as part of the COVID Genomics Consortium, we worked with Nick Lohman in Birmingham. The CLIME system really underpinned the whole of our global approach to COVID genomics with the scheme that Andrew Rambow developed and Onyo O'Toole for developing these nomenclatures. Now, these trees are really beautiful, but they're ultimately quite useless for public health. We need to be describing these things in units. So developing these nomenclature systems, alpha, beta, etc., that then became known means that we can describe a public health unit to anybody in the world and they sort of know what it is. So we were thinking, okay, so during COVID, we completely threw out our ways of building trees and started again to be able to visualize trees of this size. But actually, the best approach is to try and think of other ways to depict this information. So quite early on, when we got to this stage, we realized that sending a tree like this upwards towards the government or anywhere else is ultimately useless. So we started to represent the information in this way. So this is time, and this is the distribution of the variance over time. So this is early on in the pandemic, where you can see there's lots of different variants, nothing major frequency. And then when we get further on, this is B177, and then this is where the WHO took these nomenclatures on, which was starting to get like IP addresses, and defined these kind of nicknames, alpha, beta. And this is the proportion of those variants over time. So you can see that there's a sweep here. This is the wave of alpha. This is delta. And this is a fairly old slide. These are the first cases of Omicron in the UK. And there are no prizes for guessing what happened to this picture later. But this kind of approach for describing these units, these variants over time, gives a much more visual interpretation. And of course, in COVID, we had a virus, we had these lineages, and we had our risk markers, which were mutations of note to keep an eye on and to help reformulate the vaccines. But the same principle of this simplicity can be applied in antibacterial resistance. We have a bacteria, we have our nomenclature systems, and we have AMR determinants that are our markers of note. And when you combine this simple information with time and place, this really yields actionable information. So if you think of the pandemic, variant, mutations of note, time and place, those bits of information allowed us to understand what was where and where it was going. So in that simple framework, where are we with AMR genomically? We know there's big databases out there, huge amounts of information. 30 million SARS-CoV-2 sequenced in 3 years. Fantastic. So where are we with antimicrobial resistance? We've got all of these species defined by the WHO. We've got our INSDC databases, ENA, which store the world's genomic information. So what we wanted to do is, can we have a real-time system for processing that information, making it all available for the world in these kind of pathogen watch systems so that anybody can analyze, but also think about, can we do automated QC, the analysis, and give live situational awareness of our genomic landscape. So it turns out that over the last 20 years, there's probably about 500,000 high-quality genomes for all of those species combined, which have time and place submitted in these archives. It's not great, right? I mean, we really need to kind of up our approaches to increasing the value and increasing this information. So it's nice to have a little figure there where I can describe it, but what we also want is to have situational awareness that allows us to monitor progress over time. So I promise this is the very last thing I will show you. So some very talented members of the team spent a lot of time automating that process. So what we now have is an automatic ingest of data from the genome archive. So anytime somebody puts one of those species into the ENA, it's pulled through, QC'd, analyzed in Pathogen Watch, it's made available, so that if anybody uploads a genome sequence, they can contextualize. Back to that point of context is very important. And then those analytics, the 4 axes of information, time, place, variant, resistance determinants based on bug-drug combinations, are then pulled through to summarize here. So we can choose any of our priority species. Let's choose Klebsiella. And this now just shows there are about 47,000 high-quality Klebsiella pneumoniae genomes. This shows here the number of those in each country. This is the distribution of the genotype. So this is the equivalent of a variant for Klebsiella. And then mechanisms here. But the information is not well sampled in any way, shape, or form. But what we need to do is start being able to describe that it's not well sampled in any way, shape, or form. This is the African continent. And for Klebsiella, which is one of the highest priority WHO pathogens, any country that's this color, there's not a single genome from the last 20 years. And that information can give us huge amounts of precision to really think about rapid interventions, developing diagnostics, vaccine development, monitoring. So this kind of approach here is not draw conclusions about the quality of the data, but it gives us a really nice way of describing within that kind of WHO level a roadmap towards what's necessary to plug these gaps, both infrastructurally, but also with respect to the information that can take us forward. So I'm actually going to finish on this slide. So what we're focused on now, really, is how to kind of progress this roadmap from sort of phenotypic information in countries all the way through to understanding what's our genomic landscape and how can we fill those gaps in to help address some of the highest priorities. Can we include that with respect to prioritization versus infrastructure? So because of the work that we've been doing and everyone's doing about understanding those kind of horizontal processes vertically, we have good examples of how it can work. So what we want to do with the advantage of being linked directly to the WHO is start to think about how this can be prioritized and funded going forward towards the ability to have this kind of real-time monitoring. So just a few quite personal acknowledgements. These 4 individuals here, for me, have just really sort of been inspiring because we're sat in Oxford, you know, in front of our fancy computers. These folk here are really driving ground-level genomic surveillance of AMR. Iruka's a very inspiring individual who has really again, taken in Nigeria, written the National Action Plan and helped develop the whole of the Nigerian CDC. Sonia in the RITM, you know, getting this kind of genomic process thought through very carefully and adopted by the government. You know, there's our sustainability example. Ravi in India, very strong interest in vaccine development and actually how you use this information to show that rolling out a vaccine is effective, or are there replacements coming in, particularly strep pneumo and Klebsiella. And the One Health approach that Pilar produces. But every single one of these individuals is sharing the information across our consortium. So Iruka, for example, is thinking of One Health approaches that are being developed in Colombia. Certainly everybody's looking towards the Philippines as being a good gold standard exemplar. Khalil, I have to shout out. Khalil shares the same sort of nerdiness for JavaScript that I do, and is just an incredible engineer, and it's a real pleasure. One of my most satisfying points of the day are when we come up with another function, and I'll excitedly talk to Khalil, and it'll be there the following days. Just very inspiring. He also shares a great and strong love of Seinfeld, so that's also a good thing. And then throughout the last 15 years, really, I did my PhD with Brian. Brian co-invented multi-locker sequence typing with Ed and Martin and other colleagues. Didn't laugh in my face and tell me to get out when I said I'd like to mess around with mobile phones for a while. Just really let me kind of go, and I appreciate that much more. Always had an open door policy that I could knock at his door and have a chat about anything. That really, really meant an awful lot. Ed really fired my interest in evolutionary biology, and still continues to, and has a very strong interest in applying evolutionary methodologies to public health, especially in the field of plasmids. I think he's doing vast amounts of great work. HIO, public health, really introduced the focus on actually we can use and translate this into real-world public health. And those projects with the European antimicrobial resistance system, seeing all the national reference labs come together, very, very, again, inspiring. And Deirdre, who's agreed to spend the rest of her life with me, so I've got to thank her. Anyway, he's in the audience. Sorry, darling. And there's one last unicorn for Orla. And then really, the team. I am very, very privileged and very lucky to work with such a bunch of talented engineers and scientists, and that's what makes this job worth doing, as well as the impact that they're all having on increasing public health for genomics. So thank you all very much.